There are many bacteria found inside of your body. Among them are probiotics, which live inside of your gut and your small intestines. Normally they are stuck inside of your gut because of the gut barrier. Sometimes though, bacteria are able to escape and make it to other organs in your body, such as your liver, kidneys, and spleens. Do these translocations cause issues though? It turns out that if bacteria get into your liver, it can cause harm, which we will discuss further down with some nice animations for you. In a study by Yale University, they found that certain bacteria in your liver cause autoimmune responses, meaning your body's infection-fighting cells cannot tell friend from foe and start attacking your healthy cells. They performed this research on mice who were carriers of genes that cause autoimmune responses, but did not show any symptoms of the disease. Yale researchers injected them with specific colonies of bacteria. A control group was kept germ-free, while 3 other mice groups were monocolonized. One with Enterococcus faecalis, or EF, one with B theta iota micron, we'll call him BT, and Enterococcus gallinarum, or EG. This research team found that Enterococcus gallinarum is able to spontaneously translocate outside of the gut to the liver and cause gene expression for most of the autoimmune disease genes. They cause autoimmune responses because of this. Meanwhile, the BT and the EF cause no harm, not much, resulting in some of the mice being affected. It turns out, however, that when given a vaccine, the mice with EG were able to have their autoimmune genes remain suppressed and no harm was done to them. So that's an improvement for sure. Afterwards, Yale tested whether hybridized mice were affected by E. gallinarum. A mouse group was colonized with both EG and Salmonella and compared by other germ-free control groups. In this experiment, the E. gallinarum did not trigger an autoimmune response in the mice. In the presence of competing bacterium, the EG did not cause an autoimmune genes to be expressed. Compared to the mice in the previous experiment, this was a significant discovery. It turns out the gene expression was more complicated than just whether or not the mice had EG or not. After these experiments, Yale performed tissue sample analysis on human livers with autoimmune disease and healthy livers. In their analysis, they found that 4 of the 6 livers analyzed contained other Enterococcus species, but not EG. None of the healthy livers tested positive for any. This was an interesting observation that Enterococcus bacteria were found in humans also. To help illustrate the point, Yale performed an RNA sequence of 14-week-old mice that had been colonized with each strain of bacteria. They compared the relative gene expression of genes that cause autoimmune responses. Bright red areas showed high expression of autoimmunity, and blue areas showed none. The columns represent the 3 different bacterium and the control. The rows represent the autoimmune genes. The EG columns express these genes the most, while other columns for the BT show little expression. In the bottom right, we can see some relatively high expression for the EF genes, supporting their findings of the liver analysis. The Enterococcus causes autoimmune responses. In summary, a large factor of the experimental design was that the mice that were chosen for this study were genetically predisposed to autoimmune disease, meaning they were expressed the genes that were typically associated with systematic lupus. In Module 3 of Biology 1510, we discussed the complex process of gene expression as affected by numerous factors. Part of this is that genes express themselves differently based on where they're located. All genes go through transcription and all genes go through translation, but not all genes are necessarily expressed in the organism. Normally there are multiple forms of gene regulation that control which genes are expressed. For autoimmune diseases, the genes are recessive and require the individual to have the gene on both halves of their DNA in order to be affected by the symptoms. In the presence of E. gallinarum, organisms who are carriers of autoimmune genes, but not necessarily affected, will have their recessive genes expressed. As we can see from the rats in the experiments above, recessive genes can become an issue if intron gene regulation becomes uninhibited, leading to incorrect gene expression that may result in health complications due to recessive genes poking up and causing some trouble.